Okay. Mm-hmm. Yep. All right, Michael. I'm not sure if I lost everyone else or what happened, but um, yeah. Okay, Michael. Ready to get started? I don't see any.
Sure.
Yeah, I don't see anyone else has joined us, but you're on time, and we might as well record something for the rest of the class to listen
Okay.
to. All right. So first, I want to let's see, share the screen. My screen. Okay, good. So, so in our course, I'm just going to our current week, and we'll go look at the study guide to get started. Okay, so here's a copy of the study guide. Um, First, I'm going to just open it up to any questions that you have, and then I'll go over um, some things that I want to make sure that we cover.
Okay.
Okay?
Um, I guess first I would ask in terms of the uh, chapters covered. I know you said, and the guide says emphasis on chapter five. Um,
Yes.
that
Yep.
seems like a very strong emphasis when I am looking through the questions. Like 80-20 or something.
Yeah, it will be probably more like 65 to 75% of the material. Uh, I haven't I haven't calculated exactly the
That's
percentage, but
fine.
it's
I don't
mostly
expect that.
chapter 5. And the part of chapter 5 that I'm emphasizing is the bacterial structure and function. So there's some overlap in this chapters 4 and 5 um, as far as like the functional part. So like chapter 4 goes over how a semi-permeable membrane, like the cell membrane, works.
Yep.
Right, but then in chapter 5, you need to understand that, well, all of the microorganisms that we're studying have a cell membrane.
Right.
And then
Right.
some of them have also kind of internal biological membrane bound compartments. But they all have those properties of semi-permeability that makes it different from, say, the cell wall or something like that.
Right. Right. Okay. Yeah. I asked because I wasn't feeling great yesterday and had planned to do, more work than I ended up doing. So today I'm trying to, catch up a little bit and maximize my time. It's actually kind of funny. This is a throwback to the first exam. I felt that kind of general achiness, you get before you get sick. And I was like, oh, this is prodromal. Application.
Nice, nice. Yep.
So anyway, yeah, I'm just trying to make sure I, spend my time strategically, shall we say.
Yeah.
Um, and so I'm gonna, I think I'm gonna do chapter 5 first and spend more time getting that down pat,
Hmm.
and then see, how I feel. Then go back and work at 4 with, what time I have left.
Right. And the staining with chapter 3, I mean, we did it in lab, so hopefully it's familiar to you now.
Mhm.
And, it's emphasis on the gram staining that we did in lab um, as the as the questions kind of guide you towards, the gram staining procedure and why is it still a valuable test. So that's just a small part of the exam. It's
Mhm.
um, not going to be more than like 10 points.
Okay.
So, yeah, I think that's a good strategy is focus on chapter 5. with the understanding that some of the details you need to know about Chapter 5 as far as the function of these, the anatomy that we are learning about for microorganisms like bacteria is covered in Chapter 4.
Yeah, I think I'm going
There's a
to.
lot of overlap.
I
Yeah.
think I may end up just combining my notes. I took quick notes
Yeah.
before, so I'm kind of
Yeah, you can
reworking
reorganize.
them. I find that helps
Yeah.
for me.
Yeah.
So instead of trying to like really redo four, I'm just going to redo five, and then I can tack some stuff on from four where it seems appropriate.
Right, right. So, and the study guide questions are meant to kind of guide you um, in that reorganizing as well.
For sure.
Okay. So anything else that you want to go over first, or should I go over my materials that
No,
I want to
I
cover?
so far I don't have specific questions about topics and content.
Hmm.
I feel good based on where I'm at and having looked through the questions. There's nothing that is so foreign to me or that I can't go check in the book
Okay.
and stuff like that.
Okay, so I'm going to spend a little time going over um, some of the details that I want you to have under your belt of the three topics that we have for the open response topics. And so first, I want to go over the function of the cell wall and the cell membrane. I already did quite a bit of this in our in the lecture I gave before we started lab work last week with giving the example of how Vibrio cholera survives, this bacterial pathogen survives in its reservoir, the habitat that it has in the ocean versus like a human reservoir, and how it survives using osmolytes and using the how the properties of the cell membrane are that would normally cause the cell to shrink in a hypertonic environment like the ocean, how that is counterbalanced by these osmolytes, as well as how the cell wall is really resisting that turgor pressure um, that would cause an imbalance in the pressure from the outside versus the inside of the cell. So the cell wall is really keeping it from collapsing. Um, so let's see. Let's go back to chapter 4 material, both chapter 4 and 5, and open that up. Gina, can you hear me? This is Professor Olkin. I'm so glad that you were able to figure out how to join us. I made a mistake on the timing of the meeting. My apologies. But we just got started, so you haven't really missed anything. I just wanted to go over a couple of points about the cell membrane. So the cell membrane, whether it's in a bacterium or in a fungal cell or algae or protozoa or helminth, any of these examples of a living microorganism, not a microbe, as opposed to a microbe, the non-living members of the microbial world, will be bound at the very least on the outside by a cell membrane. So a lipid bilayer, as you can see here on the left in this image from your book. The lipid bilayer is mainly composed of phospholipids in two layers. and they're organized with the hydrophobic tails, the hydrocarbon tails, facing in toward each other, whereas the phosphate group heads, which are hydrophilic, the water-loving part, are facing the outside aqueous environment, either the outside of the cell, which is aqueous environment, or the inside of the cell, which is also an aqueous environment. Besides lipids, though, the other major um, macromolecule in the cell are proteins. And these proteins have a variety of functions, including communication within kind of detecting what's out there in the environment around it, as well as these transmembrane spanning proteins that are important for transporting um, molecules either into the cell, so like food, like glucose, or out of the cell in a regulated way. Okay, so remember that this lipid bilayer, because of its hydrophilic, hydrophobic orientation, is semi-permeable. Not everything can get in as uh, freely in and out, right? Only very small molecules that are not charged and some polar small molecules can get in. Nothing big can get in and nothing charged can get in. So but the cell at times does want to bring large molecules like sugars in for food, for creating energy like ATP. So it really has to use these special proteins that are able to help the larger molecules or the ions. The ion channels also are transmembrane proteins that help ions get in or out. And then what moves freely across the lipid bilayer are things like water, which we know the definition of the movement of water across a membrane is osmosis. All right, so that's the membrane, and bacteria have that. And this is a summary on the right of what's getting in and out of a cell, across a cell membrane. But what happens, and here on the left you can see another image from your book, what happens to a cell when it's in an environment that is not balanced in solutes. So isotonic solution or environment is balanced. The number of solutes or the concentration of solutes on the inside of the cell is the same as on the outside. So that doesn't mean that there's no movement of water. It just means that the osmosis that's happening is the movement of free water equals, that's the free water molecules that are moving into the cell is equal to the free water molecules that are moving out of the cell. But when we get to a situation where there's a lot more solute, for example, in the ocean where there's more salt, a higher concentration of salt dissolved in the water, those ions, the sodium and the chloride ions, create an imbalance with what's inside the cell, because the cell, really the function of the membrane is to maintain the condition of the cell so that it can maintain life. It can carry out its metabolism. So in this case, it's imbalanced. So the hypertonic solution means that a solute concentration on the outside is higher than on the inside of the cell. And free water molecules want to move out, causing the cell to shrink. Now, if this was a human cell in the ocean, that is exactly what would happen, because human cells don't have a cell wall and don't have mechanisms such as bacteria to kind of offset this imbalance. But in the case of Vibrio cholera, as we discussed in lab, it has two things. It has the cell wall that's very rigid that prevents the cell from dying, and then it also balances out so that you don't get the cell membrane pulling away from the cell wall, it balances out that solute concentration by producing osmolytes, or these other electrolytes that help to balance the solute concentration, to fool the cell into thinking it's balanced, even though
That's right.
sodium-wise, there's a lot less sodium inside the cell than outside the cell. Okay? Yeah, Michael?
Sorry for interrupting. You said it was the cell wall itself that produces the osmolytes, not something internal to the cell?
No, it's internal. So
Oh, okay.
there are two aspects that keeps the bacteria alive. One is the cell wall, and it's very strong and very rigid, and so it keeps the shape. That's the function of the cell wall, is to keep the shape, determines the shape of a microorganism that has a cell wall. It can be bacterium, archaea, or fungi. Um, and it also resists the turgor pressure. So for example, in a hypotonic solution, it would completely prevent the cell from bursting, right? So let's look at the hypotonic solution. That's the opposite, where a situation where the outside environment is very low in solute concentration, and so the free water would want to move from the outside environment to inside the cell, and that would cause the cell membrane to swell, and if it got swollen enough, it would break that lipid bilayer apart, and so the integrity, that semi-permeable feature of it, would be lost. And so the mixing of the outside environment with the inside of the cell would essentially kill the microorganism. But bacteria have a cell wall that prevents the cell membrane from bursting to that critical point because it resists that pressure of the water that's coming in.
So the
Um,
water can get in
it can
and
get
does
in.
raise some pressure and affect some concentrations inside,
Hmm.
but
But
only up to a certain point when that internal pressure is enough to resist further movement of water.
yeah, right, it will prevent further movement. So one side note here is that antibiotics that target the cell wall, the way they work, and we'll learn that in later in the semester, but just as a preview, um, the way they work is that they disrupt the property of this cell wall in that they no longer can resist the turgor pressure because they're breaking down that strength by breaking apart or preventing the synthesis of new parts of the cell wall. So the cell wall slowly weakens due to the drug being present. And then if the cell wall is weak, it can't keep the shape of the cell, and the turgor pressure will lyse the cell and kill the bacterium that it's affecting. Um, okay, so so let's take a moment just to look also at the cell wall, which is, I think, yeah, here it is. In Chapter 5 material, it goes over it in detail. So peptidoglycan is common between both gram-negative and gram-positive, but remember that gram-positive have many, many layers of peptidoglycan. And if you look at the image on the left, that gives a detailed structure of the peptidoglycan. Here on the very left of the image, you see the strings of sugars, so the glycans that are alternating in their form of N-acetylglucosamine and N-acetylmuramic acid, which are detailed up here in the right corner. You don't need to know the detailed structure, but you do need to know that these are many, many layers of these repeating strands of glycans, right? And the important thing are these colorful dots that show how they're connected to each other, and that gives the strength to the many, many layers. because they're covalently attached. And here in the center you can see how they're covalently attached by these cross bridges. A peptide of 4 amino acids, so a tetrapeptide, forms off of muramic acid. And these peptides then covalently link the layers, so from one layer to the next, through a covalent bond with the amino group and the carboxyl group of amino group of one amino acid with the carboxyl group of another amino acid. And it's that covalent bond. Remember, covalent bonds are the really strong ones that hold together. They take a lot of energy to break them. So it's that covalent bonding between these many layers that adds to the strength of the gram-positive cell wall. Um, let's see what else. So here it mentions that the target of penicillin actually prevents the formation of the covalent bond, and that's why it works at curing gram-positive infection, because the penicillin will it really only works when a cell is growing and dividing and making new cell wall. But once it's unable to covalently link those many layers, the cell wall weakens, and then the water will cause turgor pressure that will burst the cell membrane that's underlying that cell wall and kill the cell. And that's how you're cured of an infection by penicillin, a bacterial infection with the treatment of penicillin. In the case of the gram negatives, and by the way, we have our own body has enzymes like lysozyme that help to break down cell wall, this time by breaking the covalent bonds in between the two sugars, the NAG and the NAM. So this covalent bond is broken by lysozyme. And we have that in our saliva and mucus secretions that help just naturally protect us from bacterial infection at all time as part of our innate immunity. So let's look then at the gram-negative cell wall. That's a gram-positive. Okay. So the gram-negative cell wall on the left here includes a few layers, maybe just one or up to 3 layers of the peptidoglycan, which does give some rigidity, and it's covalently linked through part lipid, part protein molecules, lipoproteins, to a lipid bilayer that's another lipid bilayer. So this inner membrane is what we commonly call the cell membrane down at the bottom of the image here. Up at the top here, though, is what another lipid bilayer. As you can see, it looks a lot different than the cell membrane, but there's some common features. So you can see that the lipids have their hydrophobic tails facing each other. So it is a bilayer of lipids, but the lipids are not all phospholipids. So the main lipid on the outside leaf of the bilayer, of the 2 layers, is LPS, or lipopolysaccharide. And lipopolysaccharide is let's see, is it's mostly a sugar that's covalently attached to a lipid. So here in the bilayer you can see the lipid portion, and then jetting out into the outside environment is the polysaccharide, so the chain of sugars. And it's that lipopolysaccharide, or it's this outer membrane first, that gives the gram-negatives this special feature of having a semi-permeable outer layer. So if we look back at the If we look back at the gram-positive cell wall on the right of the screen here, you can see it has its many layers. Um, might even have, not in all cases, but in many cases, there's an S layer with some sugar chains um, protruding out from the last layer of peptidoglycan. Um, but that peptidoglycan is completely permeable. It's just a lattice work. Molecules can freely, large molecules, charged molecules, small molecules, uh, polar molecules, anything can get in and out. So it's not restricting any movement of molecules in or out of a gram-positive bacterium. But in the gram-negative bacterium, you have this outer membrane that is restricting. And really the important component, so just like your cell membrane, the, the outer membrane has mostly lipids, but also importantly has some proteins in it. And those proteins include transmembrane proteins of a class or a group called porins, which form a channel through which large or charged molecules can get into the cell. Then they get into this periplasmic space where you have a little bit of peptidoglycan, which is fully permeable. It's not restrictive in any way. And then it hits the cell membrane where again it has a semi-permeable barrier that it would have to get across. All right, so let's see. So that's the function of the bacterial cell membrane and cell wall, and its role in survival, either survival with antibiotic treatment or survival in the environment. I went over the difference in cell wall composition covered in Chapter 5 between gram-positive and gram-negative. I didn't go over all of the details, but I went over the most important details to remember, and how antibiotics such as vancomycin and penicillin was the example I gave, target the cell wall of gram-positive but not gram-negatives. And then external structures and internal organization of prokaryote and eukaryote microorganisms. Before I go on to that, I want to go back because I had given you a handout in lab last week, and we didn't have time to go over it. So I just want to open up to any questions you might have had from that exercise that I gave you. So Mike or Gina, did you have any questions about what's what on this diagram or in the table?
No, I didn't. Full disclosure, I saved this and didn't do anything with it yet, other than look at it.
Okay. So if you want to do it on your own, you can mute so you don't hear what I say, but you can come back and compare your answers with what I go over here.
Okay.
Okay? So the first image in part 1 is a gram-negative bacterium. How do we know it's gram-negative? Because it has the inner membrane and the outer membrane. So I tell you which side is the inside of the bacteria and which side is the outside environment, right? So you see these two lipid bilayers, and so right away you should know it's gram-negative. At the top here is part of the LPS called the O polysaccharide. For B, I'm circling the part of the lipopolysaccharide that's embedded in the lipid bilayer. So that's the lipid A portion. That's actually also known as endotoxin. Part C is a protein that you can't quite see it, but it goes all the way through the lipid bilayer, and that would be a porin protein. Part D is the lipid bilayer outside of the cell membrane, so that's the outer membrane. And then this bracket is meant to be showing the space in between the outer membrane and the inner membrane, and that space is called the periplasm. And then these alternating colors, cylinders, are meant to represent the chains of glycan that are part of the peptidoglycan that you would find both here in E in the gram-negative cell as well as here in part A of the gram-positive cell. And then finally, G, as I mentioned, is the inner membrane, or you could call it the cell membrane. So let's go to part 2. That's a gram-positive cell wall. Right away you know it's a gram-positive cell wall because it has these many, many layers of peptidoglycan, which is part A. Part B is the teichoic acid that spans from the cell membrane through this very permeable layers of the peptidoglycan. And then the dots uh, here in C are those peptide cross bridges that I said were so important for covalently linking these many layers of glycans in the peptidoglycan of the cell wall. And it gives really the strength and rigidity that you find in the cell wall. And then key differences you might have already figured out. Um, Gram-positive cell wall has very thick peptidoglycan, gram-negative thin. Gram-positive has no outer membrane, gram-negative has an outer membrane. Gram-positive has teichoic acid that's not found in gram-negative. And there are a variety of other possible answers, but another one is that gram-positive cell wall has no porins, whereas the gram-negative has porins in the outer membrane portion of its cell wall. All right, so it's really the cell envelope that technically that I'm asking you to answer here, the differences. Um, and that's all you need to know about the handout that I gave you. Okay, so moving on. Um, so I've already gone over prokaryote and eukaryotic differences in cell structure, and that is repeated in chapter 5. Importantly, let's see, I think I have those notes open. Yeah.
Professor Oakham.
Yeah.
Before you dive into this, for some of these terms and stuff like lipopolysaccharide, the N-acetyl mu, the
Uramic acid.
thank you.
Yes.
If are we okay using acronyms, or do we need to be able to like write these things out?
So I am not going to ask you to memorize N-acetylmuramic acid or N-acetylglutamic acid, but be able to recognize it. And for the LPS, actually you should know it means lipopolysaccharide, but
That's a little easier.
either one. Either one is. I think it's actually helpful to learn it as lipopolysaccharide because then you can remember that there's a lipid portion and a chain of sugar portion, right? And the lipid portion is really relevant to its function as a toxic part of the gram negatives in that when uh, when a cell dies or when it's killed by, say, antibiotic, the gram-negative cell has this built-in toxin that once it's released from the cell, from the outer membrane, it will be toxic to the host. And it's a serious problem with like toxic shock that may result as from a systemic infection by a gram-negative bacteria, so a sepsis. Like once it gets in your blood, the infection can spread to all of your tissues and all of your organs, and then you get sepsis. And that's when antibiotic treatment would not be recommended because it would just release more of the toxin, endotoxin, from the gram-negative cells and cause um, more symptoms, more severe symptoms. Um, So yeah, you lipopolysaccharide, yes. The NAGNAM, no. Any other questions about terminology?
I presume, like the prior exam, where there are instances of, you know, historical this or specific bacterium that exhibits these characteristics. That didn't seem to be a big thing last time. In fact, I remember you saying no historicals.
Oh yeah, don't worry about historicals. Yeah, I'm more testing your practical knowledge of, not like who discovered what or when it was discovered. Um, okay, so moving on to these key differences in structure between prokaryotic cells, such as bacteria, and eukaryotes. Be sure you know in and out the details of the structure of bacteria. Um, and then those major differences between bacteria and eukaryotic microorganisms, or us, our own cells, as eukaryotic cells, human cells. So here I have in this slide on the left, in the upper right, there's an electron micrograph that's been colored. It's actually a scanning electron micrograph. And the color has been given to the genetic material, the DNA, and in particular the chromosomal DNA. So as you can see, it's really concentrated in one part of these two cells. These are actually two bacteria that have been reproducing and are still slightly attached here. So that there are two red regions because each cell has its own region where its own genetic material. So, but the genetic material is restricted within the cytoplasm, but it's not separated from the cytoplasm by a lipid bilayer. Okay, so there's no nuclear envelope, there's no nucleus, but there is a nucleoid. Be sure you know the difference between the nucleus, the nucleoid, and the nuclear envelope. And DNA being the macromolecule, not the compartment. So the chromosome, as would a chromosome in our own human cells, or the chromosomal DNA in a fungal cell, the chromosomal DNA is all of that DNA that's required for the organism to reproduce itself. Turns out in bacteria there's another type of DNA that's not found in human cells, for example, called plasmid DNA. And plasmid DNA is extra chromosomal. By that I mean it's not part of that set of genes that is required for the bacteria to reproduce. So they can actually lose the plasmid DNA and they can still survive. But the bacteria that have plasmid DNA actually often keep it in their cells and copy the plasmid, which can be replicated independently of the chromosomal DNA, and pass it on to their progeny, because the plasmid DNA sometimes carries genes that give it a growth advantage, and that includes antibiotic resistant genes. Okay, so we have some appendages. Here we are at the eukaryotic cell. Know the general membrane-bound compartments. Remember, some of you didn't get this last time, but remember that ribosomes, whether they're on the surface of the ER or in the cytoplasm, are not bound by a membrane. They're simply an RNA protein complex that functions as a machine to make proteins from the message in the form of messenger RNA. Okay, so ribosomes are organelles found in every living thing. Bacteria have it, even though they don't have internal membrane-bound compartments. They have ribosomes. Um, and then the nucleus is where the genetic material in eukaryotic microorganisms or eukaryotic organisms in general are found. Um, okay. I think that's all I want to say about prokaryote versus eukaryote. There's a nice summary table here from another um, resource, another textbook that helps to highlight some of these key differences between prokaryotes and eukaryotes. Um, all right, so back to our study guide. Okay, so once again, Gina, I'll ask, or Mike, if you have any questions so far. Um, if you have any questions so far, I'll go over them. Otherwise, I think that's about what I wanted to cover.
I feel all right. I think.
Yeah, I think, oh yeah, Gina, yes.
Um, I think I feel alright too.
Oh, good. So yeah, just as I mentioned, like use the study guide, write out your open response, like as much details as you can. Go through those questions that we had for homework and try to answer them without looking at the multiple choice possibilities. So as if they were like short answer questions or fill in the blank kind of questions. Um, and just go through the bullet points that I have here for the short answer questions, and yeah, I think you should be good to go. As I mentioned, like you can see that a lot of the material is from chapter 5, but also some of so for example here where it says describe the cell membrane structure, composition, permeability, and nutrient transport. I went over almost all of that except for the nutrient transport. That you should know like how glucose, for example, gets into the cell, which is the favorite food of bacteria. Um, and permeability is gone over in chapter 4 of your book, so know how it's permeable and not what gets in and out freely through diffusion without any input of energy and what needs transport or an input of energy to get it in or out. Um, Yeah, I wrote out lipopolysaccharide here lower on the list. So yeah, be sure you know LPS is lipopolysaccharide. But as far as the other terms, uh, the N-acetyl-muramic acid, you just need to be able to recognize that, not spell it right or anything like that. Yeah, I think that's it. And just the part from lab that you'll need to know is especially the gram staining that we did last week. So be sure to review those steps and why we do gram staining as explained in your chapter 3 of your book. Anything else?
Not for me. Thanks for doing this again.
Okay, you're welcome. I'm sorry for the glitch about the the meeting being the wrong time. But hopefully everyone will find the recording and be able to get the information. All right. So I'll see you this week in lab.
Thanks again.
Thank you.
You're welcome.